It's a great pleasure to be here. I'm very happy to see so many students here today. My name is Bengt Fadeel. I'm from the Institute of Environmental Medicine, which is one of the departments here at KI. I've been some 20 years at the Institute and almost 30 years at KI. I also did my MD and PhD education here at KI. The topic of today's presentation is engineered nanomaterials. And I decided to call this the good, the bad, and the ugly. And, well, the good, by this I refer to nanomaterials in clinical use, which is, in other words, nanomedicine. And by the bad, I refer here to nanotoxicology, or the potential toxicities of engineered nanomaterials. So this is something that we have been studying in great detail, actually, over the last 10 years. Now, if you stay until the end of the lecture, I will also tell you about the ugly. Now, what are nanomaterials? A common definition states that nanomaterials are materials that are somewhere between 1 and 100 nanometers in one or more dimensions. And the point I want to make here is that as we shrink materials down into this size range, we begin to approach the same nanoscale range of biological systems. Obviously, microorganisms such as bacteria, they are by and large micrometer sized, but viruses can be considered a natural form of nanoparticles. And there are many proteins and biological machineries in the nanoscale range. So the important point here is that engineered nanomaterials are somewhere in the size range of natural biological systems. And a few words on nanotoxicology. The whole point of engineered nanomaterials and why people synthesize such materials is that we want to have materials which have novel and useful properties. So as we shrink a material from its bulk size down to nanoscale, the material properties also change. One example here is shown here is for gold, and we are looking here at the melting temperature of gold as a function of the diameter of the particles. And as you can see, this physical property of this material changes as we shrink the diameter of the particle. Now, if we accept that nanomaterials adopt novel properties, then we also have to consider the possibility that these materials may also have unexpected or novel hazardous effects not seen for the same material in its bulk form. So this is really what nanotoxicology is all about. However, that's not to say that there is necessarily a risk involved. Risk is a product of both hazard and exposure. And just to illustrate my point, here is a picture taken in an aquarium. And we have a potentially dangerous creature here, this shark. But the people observing this scenery are not exposed as such, and therefore there's no risk. In contrast, if you consider this scenario, this is definitely hazardous, again, the shark, and there is certainly exposure. Therefore, there is a risk. So when we consider hazardous materials, such as, for instance, nanomaterials, we have to consider not only their intrinsic hazard, but also whether there is a possibility for exposure. And if there is exposure, whether we can reduce or minimize the exposure. Well, again, the question is, are we exposed to particles? And the answer is yes. This picture is taken in Hornsgatan in Södermalm in Stockholm. And there are certainly a lot of particles in this environment, both nano-sized particles and larger micrometer-sized particles. And what's the source of those particles? Well, there are at least two sources of particles in this particular case. One is, of course, the particles that are generated from combustion engines. But the other is maybe more unique for this region, and that is due to the studded tires, dubbdäck in Swedish. So the studded tires, when they are when you drive your car on a dry surface, there could be a wear and tear of the studded tires, giving rise to the release of nanoparticles in the air. Furthermore, there was a paper published by French scientists only a few years ago showing that there are also carbon nanotube-like structures both in vehicle exhaust, as shown here by electron microscopy. These are the carbon nanotube-like structures, as well as in the lungs of children with asthma. So in this study, the authors have taken lavage fluid from children and examined the cells in the lung fluid obtained from children. And they found carbon nanotube-like structures. They didn't study healthy children or adults, but the authors concluded or speculated that all of us who live in cities with a lot of traffic most likely have nanomaterials, including these kind of structures, in our lungs. Just to give you one more example, and just to illustrate the point that we are in fact exposed to particles in our daily lives, this picture is taken from this review article published by Phil Democritou and co-authors at Harvard University, and it deals with metallic nanoparticles that are emitted from printers, that we have in our offices. And this is a glove box, and the first author here, Dr. Perella, is here working over here with the glove box and trying to figure out whether these printers actually emit particles, and the answer is yes. One can detect metallic nanoparticles that are present in the printer toner and emitted from these printers. Now, whether this is a health problem or not remains to be understood, but it is certainly a potential source of nanoparticles in our daily environment. There are also nanoparticles in consumer products, and I just show you some examples here in this slide. For instance, carbon nanotubes can be found in the bumpers of certain cars and in golf clubs and tennis rackets, bicycle frames, and so forth. There are certainly nanoparticles of zinc oxide or titanium dioxide in cosmetics or in sunscreen. And there are silver nanoparticles in textiles. And why is that? Well, silver ions and silver nanoparticles that release silver ions are antibacterial. So if you prefer to not wash your socks that often, go for the silver nanoparticle containing textiles. Personally, I would prefer to wash my socks, because what happens when you wash socks multiple times is that there is a leaching of silver nanoparticles into the wastewater. Of course, we also have the good side of nanotechnology, and one example is nanomedicine. And I illustrated here the 3 or 4 potential uses of nanomaterials in the clinic. And apart from this aspect, the use of nanoparticles to amplify the signal in diagnostic tests, there are, broadly speaking, 3 main areas of nanomedicine. And one is drug delivery, which as shown here. And the other is nanoparticles for imaging, for instance, as contrast agents in MRI. And the third is tissue regeneration. In particular, this area of drug delivery has received a lot of attention. The idea is to have smart nanomaterials or targeted nanomaterials that will, upon injection into the patient, will find their way to the diseased tissue, for instance, to a tumor, and thereby have a stronger effect in the tumor and less side effects in other tissues. So that is the aim of nanomedicine. Now, does this actually work? Well, I took one example here from the literature. This is a paper published a couple of years ago in Nature by American authors. And this is an interesting study, because it's not a mouse or animal study. This is a study performed in 3 patients with metastatic melanoma disease, so a skin cancer. And what the authors did was to produce nanoparticles of cyclodextrin-based nanoparticles that are wrapped with siRNA targeted towards a specific gene expressed in the tumor tissue. And they coated the particles with a polymer, which is called PEG, and they attached a ligand, which is, in this case, transferrin. And this is because many tumor cells overexpress transferrin receptors. So the idea is that if these particles expressing transferrin on their surface are injected into the bloodstream of the patients, they will find their way to the tumor cells that express the transferrin receptor. And indeed, the authors were able to show that this is what happened, and the particles found their way to the tumor tissue. And at some point after injection, the authors excised pieces of the tumor and could show that there was indeed RNA interference. There was downregulation of gene expression. And these nanoparticles are now in clinical trials. One more example, this is hot off the press, as it were. This paper was published only 2 or 3 days ago by authors from the Chinese Academy of Sciences in the journal Nature Biotechnology. Now, going one step further, these authors used something called DNA origami. One might argue that DNA really is the ultimate nanomaterial. And in this case, the authors synthesized DNA that contained, as I've written here, a targeting entity, which is a DNA aptamer targeted towards nucleolin on the outside, and on the inside, thrombin. And so when this tubular structure finds its way to the vasculature of the tumors and unfolds, it releases thrombin. And this causes, as I mentioned here, intravascular thrombosis. This leads to necrosis of the tumor and inhibition of tumor growth, which is illustrated here. So this is another example of a targeted nanostructure-mediated delivery to a tumor, or more specifically in this case to the tumor vasculature. Just to give you an example of the state of the art in the field of nanomedicine. Now I'd like to switch gears a little bit at this point and talk about carbon-based nanomaterials. And why carbon-based nanomaterials? Well, these are materials that are produced in large quantities worldwide, and that means that there could be a potential for exposure in the occupational setting. The workers who produce the materials are possibly at risk of being exposed. There are also studies suggesting that carbon nanotubes, for instance, could be used for biomedical purposes. So these are altogether interesting materials. However, there is a concern that certain carbon nanotubes might have asbestos-like properties. And in particular, this paper from a group in Edinburgh, which was published about 1 decade ago, suggested that if carbon nanotubes are injected into mice, they cause so-called asbestos-like pathogenicity. What do they mean by this? Well, basically they show that long carbon nanotubes triggered inflammation in mice, whereas short carbon nanotubes were much less inflammatory. And this reminds us of this pathogenic fiber paradigm, which is a well-known structure-activity paradigm in toxicology, which states that a fiber that is both long and thin and biopersistent is likely to be more pathogenic than other fibers. There are additional studies on long carbon nanotubes. This is a paper from a group in Helsinki showing, again, that the long carbon nanotubes trigger more inflammation, in this case through inflammasome activation, than the short carbon nanotubes. And based on these studies and several other studies in the literature, the International Agency for Cancer Research concluded a few years ago, in 2014, that certain multi-walled carbon nanotubes should be considered as potentially carcinogenic for humans. Whereas for other single-walled and multi-walled carbon nanotubes, there's simply not enough data to draw such conclusions. Of course, this study is not based on human data. This is an extrapolation from animal data to the human situation. But it may appear to be quite trivial, but it is nevertheless important to understand that carbon nanotubes come in different shapes and sizes, and long carbon nanotubes certainly are more pathogenic than short carbon nanotubes. Furthermore, the same Edinburgh group published a paper last fall, the reference is at the bottom of the slide here, showing again that long carbon nanotubes actually replicate the pathogenesis of asbestos-induced mesothelioma. Now, many of you may know that after exposure to asbestos, there is a long latency period. It can take 20, 30, or up to 40 years before the development of a particular type of malignancy called mesothelioma. And it has never really been well understood why there is such a long latency period. Now, in this study, the authors show that both for long asbestos fibers and for long carbon nanotubes, the possible pathway leading to malignancy. So there is, first of all, an inflammatory response and a chronic inflammatory response. And this, in turn, leads to hypermethylation of certain genes. Hypermethylation can, in many cases, cause silencing or downregulation of genes, and in this case, transcriptional silencing of tumor suppressor genes. And this, in turn, drives the malignant process. So we should avoid inhalation of long carbon nanotubes. This is certainly clear. But other carbon nanotubes, properly functionalized and so forth, may be still useful in nanomedicine. One more aspect which I think is important in this case when it comes to carbon nanotubes, we and others have shown that carbon nanotubes can be digested by the immune system. If you remember, the pathogenic fiber paradigm states that materials that are long and thin and biopersistent are more pathogenic. But this is, while this is true for asbestos, which is not degradable, carbon nanotubes can be degraded by this enzyme, which is called myeloperoxidase. And this is an enzyme that is abundantly expressed in neutrophils, in white blood cells. And this is an in vitro experiment in the test tube. And these are Raman results from actual neutrophils which have been exposed to carbon nanotubes. And we see characteristic changes of these spectra, which are a sign of degradation of these materials. So not all carbon nanotubes are necessarily asbestos-like. And to summarize this in schematic form, so there seem to be reciprocal interactions. What do I mean by that? Well, carbon nanotubes, certain carbon nanotubes may trigger inflammation. On the other hand, inflammatory cells may also strike back and degrade the materials and thereby reduce inflammation. So nanotubes may interact with cells of the innate immune system, such as macrophages, neutrophils, and others. And these cells may release inflammatory factors, IL-1 beta, TNF alpha, and other cytokines, which drive inflammation, in this case in the lungs. On the other hand, some of these cells, neutrophils in particular, express enzymes such as the myeloperoxidase, which can degrade the nanotubes, and we have shown in animal studies that this then results in less inflammation. So there is a way to control the biological fate of these materials. In the last few minutes, I'd like to touch on another type of carbon-based nanomaterial. This is graphene. Graphene is a material that has received a lot of attention in recent years. There was a Nobel Prize awarded for the discovery of graphene not too long ago. And graphene can be considered a two-dimensional material. So it is an atomically thin material. Basically, you have to picture a single layer of carbon atoms in a flat structure. Carbon nanotubes, on the other hand, are essentially a single layer of carbon atoms rolled into a tube. So it's a hollow tubular structure. But graphene is a flat two-dimensional structure. And the question that we asked is, can the immune system recognize a two-dimensional structure? And the answer is yes. And we see some evidence for this here. These are electron micrographs taken in our lab of primary human macrophages. This is a normal macrophage. This is a cell nucleus. And this is a macrophage that has been exposed to graphene oxide, so atomically thin, but several micrometers in lateral dimensions. And the macrophage has no problem whatsoever in terms of ingesting this material. And we also don't see any toxicity. We studied this further, and we found that the graphene oxide, if it is a pure material and not contaminated with endotoxin, then the graphene oxide doesn't kill the macrophages, and it also doesn't trigger any pro-inflammatory cytokines. We studied both small and large graphene oxide, and we looked at TNF-alpha, IL-1 beta, and there's hardly any secretion of these pro-inflammatory factors. So this is all very good. Macrophages are supposed to eat stuff, and they're supposed to take care of cell debris and particles and so forth. And evidently they can also engulf graphene oxide. There are other phagocytic cells, and my favorite phagocytic cell is the neutrophil. And because the neutrophil, which is often considered to be a pretty stupid cell, is actually a rather clever cell and has many different ways of handling microorganisms. So in addition to engulfment of microorganisms with intracellular ROS-mediated destruction, neutrophils can also produce something called nets. And these are extracellular structures consisting of nuclear chromatin that is emitted from the cell. And this, furthermore, these nets are decorated with granule proteins, including myeloperoxidase and other enzymes. And so this is a way for the cell to capture and degrade microorganisms extracellularly. There's a very nice review by Papagiannopoulos on this topic in Nature Reviews, just published very recently. So we asked the question, can graphene oxide trigger nets in primary human neutrophils? And the answer is yes. So in contrast to macrophages, if you recall, I mentioned that when graphene oxide is incubated with macrophages, we don't see any cell death. The macrophages just eat the material. But when it comes to neutrophils, there is a dose-dependent loss of cell viability. We studied both small and large graphene oxide sheets. There's also an increase in the production of nets. This is measured here by an enzymatic assay, and we can also visualize the nets here by staining for this enzyme Ne. So what we see here is that for the large and small graphene oxide, we do see production of nets. And we also visualize this by scanning electron microscopy. So here is a normal neutrophil. And here's a neutrophil that has been incubated with graphene oxide, large graphene oxide sheets. And as you can see, the cell produces this neutrophil extracellular trap, which is more or less like a spider web consisting of nuclear chromatin, and captures the graphene oxide. This is a cell that has encountered small graphene oxide. We call these fuzzy nets because the nets are, instead of naked DNA fibers, we now have DNA fibers coated with multiple small sheets of graphene oxide. And we also found, although I haven't shown you the data in this slide, but we also found that the GO could be degraded in nets, much like carbon nanotubes, and incidentally much like microorganisms. So based on the results that I showed you, our understanding, or let's say working hypothesis, is that carbon-based nanomaterials may actually be sensed by the immune system as pathogens. And this means that the effects are maybe not novel as such. The immune response to pathogens is an ancient response. And so if we can understand how the immune system handles nanomaterials, this will also enable us to design nanomaterials that can either evade the immune response in order to reach tumors or other target tissues, or we could design nanomaterials that can exploit the immune response, for instance, for vaccination purposes. So to summarize, we are constantly exposed to nano-sized and micrometer-sized particles, and I would say that there is a particular concern when it comes to occupational exposure, although we are also, all of us, exposed to particles as consumers as well. Then we have the particular case of deliberate exposure in the clinic. There are already nanomaterials in the clinic, clinically approved nanoparticles, and there are many more in the pipeline in clinical trials and so on. Now also mentioned or discussed nanotoxicology, and basically, in a nutshell, nanotoxicology is a scientific discipline that seeks to understand the structure-activity relationship of nanomaterials. So what are the properties that drive toxicity? And I gave you one example. The fiber-like dimensions of carbon nanotubes are mainly responsible for the toxicity of those materials. But for other types of nanomaterials, other properties may be more important. Then there is something that I usually refer to as the nanomaterial paradox, and that is to say that the very same properties that could be potentially useful in the clinic, the small size, the surface reactivity, and so forth, may also drive toxicity. So this means that we really need to understand the material properties and how materials interface with biological systems. Finally, the ugly truth. Well, the ugly truth is that despite all the research that has been going on in the past decade on nanomaterials and nanotoxicology, we still don't know very well how to regulate these materials. The risk assessment of these materials is lagging behind, and we need new approaches. I think the problem is not the lack of research. There's a lot of research. I think that there is, on the other hand, a need to have a dialogue between scientists and other stakeholders. By stakeholders, I mean, for instance, regulators or industry. And in Sweden, we have something called SweNanoSafe, which is a national platform that has been initiated in order to promote the dialogue between the different stakeholders. And I'm chair of the expert panel of this platform, and we are now building a website so that we can disseminate current knowledge on nanomaterials through this website. Finally, I'd like to thank some members of my lab, in particular Surav, who is a postdoc in my lab, and mainly the main driver behind the work on graphene in recent years, together with Beatrice, a graduate student in my lab, who has contributed also to the work on nets. And of course, our funding, we have been funded to a large extent through the European Commission. Thank you very much.